Hi everyone, my name is Kush Narang and I'm an undergraduate at UC Davis in California. And today I'm really excited to be here presenting my talk on HaloClass, which is a salt tolerant protein classifier that we've created that uses protein language models. So, obviously, being in the digital agriculture talk right now, I think it's really important for us to discuss why salt tolerance is such a relevant issue and challenge in agriculture. And for me, it boils down to two main reasons, which are symbiosis and proteins. In terms of symbiosis, there are thousands of salt tolerant bacteria and archaea that are well understood to play important roles in helping plants survive and thrive in high salinity conditions. And that's especially important when we consider the fact that across the world, soil salinity is increasing. And that poses a major challenge to the world's food supplies, and it's something we really need to think about. So, being able to identify these symbiotic organisms would help us more effectively support our plant populations, right? At the same time, the proteins and enzymes themselves within the plants and within the crops can be engineered to increase salt tolerance because they play such important roles in key metabolic functions. So we think it would be really cool if there was an algorithm that could assist in that process of identifying and even engineering salt tolerant organisms and proteins. So, to broadly discuss salt tolerant proteins a little bit more, the reason why we're able to identify them is because salinity plays a really important role in protein folding. Protein folding is an incredibly delicate and intricate process, and the salinity of a solution where the protein is in or where the organism lives plays a very important role in that protein's composition to ensure that it's able to fold. So, an effective algorithm must be able to generalize well to brand new sequences that it's never seen before, whether that's a newly designed protein that we want to test, or if that's looking at something like a bacteria or archaea that we've never seen before. At the same time, this algorithm needs to be sequence based only, since it's really rare for these types of protein structures to actually be solved as crystal structures. Understanding this, we think that any algorithm needs to function at three fundamental levels of understanding. The first is the species level. In the case of this task, it should be able to identify whether a symbiotic organism, like a bacteria or archaea, is salt tolerant. Next, on the structure level, in both a plant or a symbiotic organism, we should be able to differentiate between highly similar protein structures, like the ones we have superimposed here in blue and pink, and we should be able to tell which one is salt tolerant and which one isn't. And finally, at the mutational level, ideally, if we're trying to engineer these brand new enzymes for proteins and plants, then we want to be able to tell the effect that a single point mutation on a protein has to the overall protein stability in a high salinity solution. Understanding this, we wanted to compare against the existing state-of-the-art models for this task, which were published by Zhang et al. In 2013 and Nath in 2016, respectively. Neither of these algorithms has a true framework for assessing these three levels of generalizability, so a further objective of our paper is not just to try and create a new algorithm, but also to benchmark these existing approaches more rigorously. Broadly, in terms of our algorithm selection, we identified two possible approaches for improvement. The first was dataset selection, and the second is algorithmic architecture. In terms of dataset selection, both the past Nath and Zhang state-of-the-art models were trained on the same Zhang dataset. As we can see in this table, that dataset only had 2 organisms and 278 sequences. We argue that that's not nearly enough for the algorithm to learn the general features of salt-tolerant proteins. So to build on that, we created a much more diverse dataset that uses proteins from 14 different organisms with more than 20,000 sequences total. And we split our dataset into 3 groups for training, testing, and evaluation. In terms of architecture, both the Nath and Zhang models use manual feature extraction motivated by human understanding. What that essentially means is that a computer obviously doesn't have an intuitive understanding of what a protein is, so we need to give it some numerical constructs and values to hold onto to create a representation or an idea of how a protein looks or behaves. In this case, the representations used by the Zhang model calculate the percentage composition of each amino acid in the protein and just create a 20 feature long vector or 20 number long vector and represent proteins just like that. Building on that approach, the NAP representation uses 762 features, which also adds on dipeptide counts, isoelectric point, bulkiness, and other general properties about a protein structure and sequence. However, we think that those approaches might be too limiting. So we wanted to take a new approach that's inspired by protein language models. ESM2 was a protein language model released by Rives et al. In 2021, and we use representations from this algorithm to create our features or representations for our algorithm. In this case, this figure here is a demonstration of how a protein language model works. Essentially, a protein language model takes a protein sequence as an input, but with some of the amino acids hidden or masked out. And then over many days and even weeks of training, the algorithm goes through these sequences billions of times, learning how to replace a hidden amino acid with the correct amino acid that belongs there, just by using the context of the neighboring amino acids and the entire rest of the sequence. As it turns out, in the process of doing this iteration over and over, the algorithm creates a vector embedding or representation of a protein that is actually highly meaningful. And these representations are known to encode for secondary and tertiary structural qualities of proteins, even though they are learning from just sequences. Understanding all of this, our algorithm is motivated by the power of these representations. We take representations from ESM2 and pass them into an SVM model, which is a type of machine learning algorithm, and then we use the SVM model to create predictions for the salt tolerance of any given protein. For our aforementioned dataset of more than 20,000 sequences, we dedicated 90% of our sequences to training, 5% to evaluation, or essentially optimizing the algorithm, and then 5% for testing. First, we're going to evaluate these different approaches on the species level. In this case, we're going to start with the Zhang et al. Dataset, which, as we've already discussed, is 278 sequences from just 2 organisms. A critical note is that because the NAP and Zhang models were trained on this dataset, they have seen all of these sequences before, whereas HaloClass has never seen these sequences before. In theory, this should give the old approaches an advantage over HaloClass, but despite that advantage, HaloClass is able to score 97% accuracy, compared to 86% for the NAP approach and just 60% for the Zhang approach. Next, we used our new test set with around 1,200 sequences to compare on a more equal playing field. This is a more equal playing field because none of the algorithms have seen these sequences before, making it a fair fight. In this case, HaloClass is able to score 98% accuracy, compared to 80% for the NAP model and only 65% for the Zhang model. What this and the past slides suggest to us is that HaloClass likely does a better job generalizing to brand new protein sequences that it's never seen before. At least, that's what we've discerned from the tests on these 2 datasets. Moving forward, we were interested in evaluating all the algorithms on their structure level performance. And a great dataset for that is the Siglasoli et al. Dataset from 2009. This is a dataset of 8 pairs of salt-tolerant and non-salt tolerant homologous proteins. All of these are superimposed on each other in this slide, with the pink structure being the salt-tolerant one, and the blue being the non-salt tolerant one. In all of these structures, the lowest RMSD between pairs is just 0.7 angstroms. The average is 0.9 angstroms, and the maximum is 1.2 angstroms. Despite these highly similar structures, HaloClass makes just 1 classification error and no ranking errors, which allows it to perform better than the past approaches on this dataset. Interested in how these algorithms were able to work, or rather, how HaloClass was able to distinguish between highly similar structures, we selected this protein, which is among the most common catalysts and enzymes used in industrial processes. Here, we zoomed in on an alpha helix from the surface of these proteins. Again, the pink is the salt-tolerant protein, and the blue is the non-salt-tolerant protein. What we see is that smaller non-polar amino acids in the original non-salt-tolerant protein have been substituted to become polar and highly charged amino acids here in green and here in dark blue. This suggests to us that even when the structures are highly similar, HaloClass is able to look at the side chain identities, or the identities of individual amino acids, to distinguish between highly similar proteins. Next, we wanted to finally assess HaloClass and the other approaches for their mutation level performance, or seeing if they can identify the effect of a single point mutation or a few point mutations to determine whether there's an effective application for protein engineering tasks. Here, we used experimental data from Tadeo et al. In 2009 that experimentally tested 9 different mutants of salt tolerance for one wild-type DNA ligase 1 protein from a halophilic bacteria. This protein is 104 residues long. Here we can see alpha-fold structures for the wild type and the mutant type, with an RMSD between them being less than 0.5 angstroms, indicating this is a highly difficult challenge. What we see is that we'll consider each type of mutation on its own. First, we'll consider the charge-preserving mutation that doesn't change the overall side chain charge, but changes the length. Here, going from a glutamic acid to an aspartic acid should ostensibly increase salt tolerance according to biophysical knowledge of less bulky residues being more salt tolerant. That's exactly what we see. For all the mutants tested, experimentally, this mutation increased salt tolerance. HaloClass accurately predicts an increase in salt tolerance, and the NAT model is not specific enough to predict any changes. Next, we consider the exact same mutation but going the other way, going from aspartic acid to a glutamic acid, increasing side chain length. Here, what we find is that there are experimental decreases in salt tolerance, which matches what we find from HaloClass, and the NAT model once again shows no change. Finally, we'll consider a length-preserving mutation, here going from an arginine to a lysine. What we see here is that this decreases experimental salt tolerance, which we accurately predict with our model, and NATS doesn't predict a change. And actually, here is the final mutant, which is going from a serine to a lysine. Experimentally, this decreased salt tolerance, HaloClass accurately predicts the decrease, and the NAT model is not able to predict the change. Overall, our conclusions are that our algorithm establishes a new state-of-the-art for salt tolerance protein classification, and we've shown that on the species level, structure level, and mutation level. Future work should consider exploring fine-tuning, which is essentially another approach to train these types of algorithms to make them even more effective, or should use larger data sets to see if we can discern more concrete insights. In terms of acknowledgements, I'd like to thank the society and organization for allowing me to present. I'd like to thank Dr. Simon Chu for being my primary mentor in this project, along with the help of Dr. Knapp and Dr. Hemstrom. And I'd also like to thank Dr. Zhang at UC Davis and Dr. Yarvoy's lab, including Dr. Yarvoy himself, Diego, and Dr. Nguyen. Thank you so much. I really appreciate your time. Thank you.